My name is Jaana. I'm faculty here at UCLA in the Department of Medicine, Division of Cardiology. Here's my email if you want to reach me. Be aware that the last A is missing from my email because it doesn't have enough characters. So many times I don't get emails because people say it's Hartiala at UCLA edu. It's not. It's Hartial. So just in case. Yeah, I know. I know. So today I'm going to talk about GxE meta-analysis for CAD and especially the lessons we've learned using Biobank and all of us cohorts. So I'm going to use our GxE study as a frame so you understand kind of what we're talking about. But the core message is that identifying gene-environment interactions for CAD using meta-analysis in, let's say, UK Biobank and all of us, it requires overcoming significant hurdles in harmonizing the phenotype definition, inclusion or exclusion criteria, crucial covariates, and appropriately account for ancestry. So let's get started. There is a complex network of pathways underlying the development of CAD. Throughout the decades, EPI studies and research have identified several key risk factors associated with atherosclerosis and coronary artery disease, such as lipid levels, hypertension, inflammation, and others. These traditional risk factors are strongly supported by genetic studies using common and rare variants. However, even after identifying hundreds of genetic loci associated with CAD risk, the underlying mechanism driving these associations remain largely unknown. It's of course possible that additional common or rare variants remain undetected and could be identified with ever-increasing sample size, think infinite sample size with biobanks. But another possible explanation could be gene-environment interactions, which are not largely studied for, at least for coronary artery disease. And one such environmental factor could be, for example, air pollution, that I'm going to talk about more today. Did I not? It's not working. Got stuck. Okay, so as a proof of concept, we have this project in our group. It's called Gene-Environment Interaction between the TRP4 locus and air pollution exposure, increased risk of coronary artery disease. So this paper is under review currently, and the lead author, James Hilser, is sitting in the audience. I don't have a nice picture of James, but you can see him in person. I'm sure that all of you know James by now. He's responsible of generating everything. So how did we go about finding GxE for CAD? So first we tested air pollution associations with CAD in the UK Biobank. And here we are, I'm showing the air pollution levels we acquired. So we are using PM2.5, PM10, NO2, and NOx. We have a suspect which could be the best candidate, but since the air pollution levels are available, we can't really ignore the other air pollutants. So yeah.
So you have measurements from what, where?
It's zip code. Yeah. I have a at the very last, I have a description how the air pollution levels were measured in the UK Biobank. I will go to that if we have time, but it's based on zip codes, yes.
But is it part of the UK Biobank?
It is part of the UK, yeah. They have measurements for 2010 levels. Yeah. Also, it's notable that it's actually good time frame because this paper has been in the works for a long time. So we suspected all kinds of things, and we wanted to know that did the air pollution levels stay stable throughout the years, because if they fluctuate a lot, then one cross-section or 1 year, it might not be a good representation, but they actually stayed fairly stable, except they started to go down slightly in 2020. So we wanted to match our phenotyping also, that phenotyping and air pollution levels kind of match each other, that it will stay constant throughout the study period, if that makes sense. So we suspected for example, PM 2.5 could be a good candidate. And if you look at the mean values here, it's 10 micrograms per cubic meter, and standard deviation is 1.1. I'm mentioning this because we are going to use 2 standard deviation air pollution levels for all these pollutants. So you will get a little bit of idea what type of information are we talking about. And then size matters. PM 2.5, which is the less than 2.5 micrometer in diameter, which is 20 to 30 times smaller than width of your single hair. These are the most dangerous, as they can penetrate deeper into the lungs and even the bloodstream. So we wanted to test the associations with air pollution levels with CAD in the UK Biobank. So on the left-hand side, we have a single pollutant model. What it means that we tested each pollutant separately in a model where we adjusted for the covariates. Here we use age, sex, Framingham ATP risk score, education, BMI, and PC 1 through 20. So it appears that PM 2.5 is the most significantly associated air pollutant, but there is signal from NO2 and NOx too.
I would think that if a neighborhood's polluted, it's going to have a high level of all of these. So,
of course.
It seems as though they're different enough that some places have high PM 2.5 and others NO2.
That is possible. We just basically wanted to pinpoint that if we are going to do the GxE, we want to do it with what single pollutant and which would be the best candidate. But you are absolutely right. I mean, you don't have areas where you only have PM 2.5, of course, but the levels can fluctuate. Yeah. But then on the right-hand side, we have the multi-pollutant model, where we included all the pollutants in the same model plus the same covariates. So as you can see, the PM 2.5 persists. The effect estimate actually goes up. The NO2 and NOx are no longer super significant. Maybe they're canceling each other out. But what I'm trying to show here, that the best candidate for us in this project would be to use PM 2.5 levels. And another way of demonstrating this association is to bin the air pollution levels into quintiles. And aside from PM10, there seems to be increased risk with CAD with increasing quintiles of the air pollutants, and especially with the PM 2.5 in quintile 5, where the odds ratio is 1.21. And then we have another piece of evidence that PM 2.5 is linked to heart disease risk is our previous paper, where we show that PM 2.5 levels increase the risk of CAD severity and incident myocardial infarction in the Ohio residents from the Biobank cohort. Biobank is kind of our in-house little biobank. It consists 10,000 patients from the Cleveland Clinic. This is a heart specialty hospital, so most of these people do have heart disease. So I think 85% of the cohort has heart disease. We have genotypes for roughly 4,500 people. We also have air pollution levels for this cohort.
So when they say ambient, does it mean in their house?
In the air, basically. It's just a fancy way of saying outside. Yeah. It's yeah. It's a bit.
So it's kind of the average air pollution in that area.
Yeah, that's also based on the zip codes. Yes. And then
Did they adjust for kind of socioeconomic status?
this is a single site, and it's a specialty hospital. So
So okay,
yeah,
this definitely. But in your case, did they adjust for education? Yeah.
we will. I'll talk about that, especially with all of us. Yeah. So our findings are not unique. Decades of epidemiological research confirm a link between PM2.5 and increased risk of CAD. For example, inhaled PM2.5 can cause lung inflammation through oxidative stress. It can directly enter the bloodstream or cause indirect effects via gut microbiome, all of them leading to increased cardiovascular disease risk. However, only few validated gene air pollution interaction studies for CAD have been reported. So hopefully I have convinced you now that a PM2.5 is a good candidate to test for GxE. So what about the genetic variants? Which one should we use? So we chose to use previously identified CAD risk loci, which is approximately 320 independent loci. So we took the data from the latest and greatest meta-analysis for CAD in over 1 million participants and curated the loci and chose the lead SNP of these loci, which I now regret a little bit after hearing the Susie and Sushi talks, but that's the approach. And then we tested the GBI in UK Biobank and GeneBank cohorts, followed by fixed effects meta-analysis. So we went back to using the GeneBank cohort because we wanted to increase the power to detect the GBI. Since we had the data readily available, we wanted to use both of them. And we identified significant GBI in chromosome 15. So of the 320 loci, 16 showed nominal GxE significance, meaning p-value less than 0.05. But this is the one locus that remains significant after multiple correction. So the p-value for interaction here is 1.2 10 to the negative 4. Those who are familiar with GxE studies, this is not a modest signal. It's pretty good, right? It's not genome-wide significant, but we tested 320 loci. So if you look at the figure, it's showing that PM2.5 exposure increased the risk of CAD more among those who were carrying 2 copies of the risk allele A versus those who carried only 1 copy. And in contrast, PM2.5 was not associated with CAD among those carrying 2 copies of the G allele, which could be a protective allele. I'm running out of time, so I'm going to skip a couple of slides. So we also functionally validated this locus. This locus included multiple genes, so we wanted to kind of pinpoint which one could be the culprit. And borrowing the wise words from Nick, it looked great. So trust me, we did the functional validation even more than shown here, but I'm going to skip that. So everything's cool. So what's the problem? The reviewer wanted to have a replication cohort. So we started to scratch our heads, what do we do? Because it's not easy to get a biobank level replication cohort, especially if air pollution exposure data should be available. So James was already using all of us cohort, so we decided to take a stab at it. And this is kind of the main focus of the talk. Hopefully I can go through all the slides. So as you know, all of us is US-based multi-ethnic biobank with short-read genome sequencing with electronic health records and questionnaire-derived phenotypes. But at the time of this analysis, air pollution levels were not available. So we're like, okay, What do we do now? So curating all of us was very painful, asked James, but I think we got somewhere. So I wanted to share the kind of steps that we took. But just to illustrate the issue with all of us and CAD, I'm showing a Manhattan plot for CAD published in 2023. As you can see, it doesn't have any significant findings, not even a hint. Um, I, this is not our. I think this is 100,000 people. Yes, this looks bad, but the consensus in the field, at least CAD field, is like all of us is crap, don't use it. But we didn't have a choice, so we're like, okay, let's dig deeper. But this looks bad, right? There's no promise finding G/E if the GWAS for CAD looks like this. So then another example is this is up from our study. So this is a Manhattan plot for MI, which is a subphenotype for CAD, and this is in UK Biobank only. So we found 31 significant loci in the UK Biobank alone. So clearly there is something severely wrong with the all of us data. So we don't have air pollution levels. The phenotyping is potentially. I'm gonna go there. Yeah. So we thought that there has to be publicly available air pollution levels. So we took the data from NASA, and this is overly simplified sentence, and combined those with all of us subject with using the 3-digit zip codes. They don't have all the zip codes, the entire zip code available. So we were a little bit worried that maybe it's not granular enough, but we map the PM2.5 levels using the estimated PM2.5. Sorry, we mapped the density of the PM2.5 levels after mapping them to the 3-digit zip codes, and it looks remarkably the same than if you took any of the PM2.5 maps across the US. So we thought that maybe this is okay, maybe it's not, we don't know, but it looked pretty good. Then the first steps or missteps we did with all of us, we included all study subjects in a combined analysis. This was not a good idea. But what we learned from there, that a genetic association based on GWAS with CAD was lacking. I'm not talking about the published data, but our own data. We were like, what is going on? Why can't we find anything? This is a larger data set. And also the association of air pollution or PM2.5 with CAD was barely there, and we should have seen something. It's pretty robust association. So we thought, okay, what's going on? And who are these, all of us participants? How can we kind of compare to UK Biobank? What do we do? So is there an ascertainment difference or diagnosed done differently than in the UK Biobank? And other clear differences in exposure based on ethnicities? What about socioeconomic status? It's probably more diverse in the US than it could be in the, for example, in the UK Biobank. So how can we harmonize the data to better match with the UK Biobank and GeneBank? So since this is a multi-ethnic cohort across the US, we started to dig deeper, and we're trying to figure out, for example, the social determinants of health, and if they can play a significant role in the all of us study sample. We were trying to think similarities and differences between all of us and UK Biobank participants in terms of SCS, ascertainment, and if air pollution exposure differs based on ethnicity in all of us. So on the left-hand side, I have the same map density of PM2.5 levels. I circle the sort of the most polluted area. And then on the left-hand side, the blue shade maps, that's the proportion of all of us participants stratified by ethnicity. And I have the same area kind of marked. I think that the starkest difference is if you look at the European proportion versus African proportion. This is not representing the entire US population, just all of our subjects that we had in the analysis set, but most of the African American subjects were living in that circled area, which is more polluted. And although there are a lot of European ancestry participants all over, but there is all lighter shade of blue in that more polluted area. So there seems to be a difference based on the air pollution levels on ethnicities. So the next steps with all of us were as follows. We initially ran a GWAS for CAD in 220,000 participants, adjusting for age, sex, and 16 PCs. The 16 PCs were available, so we used all of them. So like I said before, effect estimates were weaker than expected. So we first thought, is the case definition off? So we applied our in-house case definition based on the ICD-10 codes. It was helping some, but not dramatically. Then we used myocardial infarction as a stricter phenotype. It wasn't really helping. Then we adjusted for additional covariates, smoking, different ways, deprivation index, income, education, wasn't helping. Then we restricted the analysis to self-reported whites. It wasn't helping. We couldn't see the signal that we were expecting. Then we restricted to those with CAD diagnosed before 2020. This is because of the COVID has a very strong effect on CAD risk. That was helping. But also we stratified by genetically predicted ancestry, which is part of the data, James, right? So there's a variable called pred ancestry that was helping a lot. So all these iterations is very expensive if you keep running a GWAS after GWAS. So we used a chromosome 9, and if you see above, that's the tallest peak is in chromosome 9, to test the different models after initial GWAS. But we did some additional optimizing because it's not just the genetic association. We have to build in the air pollution variable too. We kept our in-house definition for CAD based on the ICD codes, and you can see it there. I'm not going to go through all of it. Then we excluded asthmatics. PM 2.5 has a very strong association with asthma, so we didn't want to mix the effects, but this is the same approach that we did with the UK Biobank. We excluded asthmatics altogether. And then we included only those with health insurance and who own their own home. So we were attempting to control issues with arising from SCS and harmonize all of us with UK Biobank and GeneBank prior to meta-analysis. So this is not necessary maybe from some other studies, but it was helping us. We used 2 standard deviation PM 2.5 levels, and we centered on the state mean. This is a similar strategy than we used with the GeneBank. If you remember the distribution of PM 2.5, it varies greatly from across the US. And then lastly, we stratified genetically predicted and by predicted ancestry, followed by meta-analysis in all of us. And look, yeah.
Following, what does it mean? You have the PM 2.5 method.
Mm-hmm.
So for a group of people, you take standard deviation?
We calculate the state mean first, and I have state mean and a standard deviation. So I subtract the state mean, and then I divide it by 2 times standard deviation, and then use those levels to test. So basically, the effect estimates you get is for the 2 standard deviation difference, which in UK Biobank, for example, for PM 2.5, that will be 2.2. 2.2 units increase in PM 2.5 levels. Well, UK Biobank, we centered on the mean, but we didn't centered on a state mean or regional mean. But in the US, which is very large, and there is so much variation. We did this both in the gene bank and in all of us.
Then you divide it by two standard deviations per year. So when you do the meta-analysis, you do meta-analysis at the effect size or the group score?
Fixed effects, but we use 2 standard deviation for all the cohort, UK Biobank, Gene Bank, and all of us.
So
It's yeah, they're all scaled the same. Yeah. So first, we wanted to, of course, test the association with PM2.5, and just very quickly on the left-hand side, we have model 1 where we only adjusted for age, sex, and PCs. There seems to be a robust association with Europeans and African Americans. The Amerindian Asians, if you remember the map, they lived in less polluted areas, and the sample size is kind of modest, but with the combined meta-analysis, it's still significant. And then the model 2, which is the fully adjusted, including age, sex, and Framingham, ATP risk score, education, BMI, and PCs. The European association persists. The African, that goes away. This could be due to the actual, the covariates are affecting, but if you look at the sample size, it's like half from the model 1, because unfortunately we didn't have all the variables that are needed to construct the Framingham ATP risk score in all the subjects in all of us. So the meta-analysis results are fairly similar. So we thought that a model 1 is sufficient for this purpose. I'm going to go over a couple of minutes, but we studied late. So what we learned, all of us can be used for CAD or binary outcomes, but you need to proceed with caution. So this is probably less issues with continuous variables, for example, measured lipids and such. You need to consider carefully inclusion-exclusion criteria for each study, and this can, of course, be outcome or exposure dependent. You need to evaluate appropriate covariates and don't just stick with the usual suspect. Think social determinants of health, especially with chronic diseases. And then ancestry stratified analysis is crucial. This is also very informative. You might find something that you didn't even think about. And then you need to consider geographic differences, especially in our case when we talk about air pollution as an exposure. And then validate, and then you can run your analysis. So this is an ongoing effort, and for example, all of us has released new approach to phenotyping, which we will need to evaluate before applying that to the future projects. And then thank you for all the people at UCLA, USC, Cleveland Clinic, and University of Virginia who were part of this study. And I underline James, who has done all the analysis we showed today. And then lastly, we have a postdoc position open in Alai Lab, so you can either contact me or our group leader, Homan. Thank you.